So we are going to go to the last speaker. Dr. Uday Saxena is the co-founder and chief of Reagene Bioscience. He was mentor in residence and professor of translational research at Dr. Reddy's Institute of Life Science, as well as a co-founder of a startup biotech company. He has held executive and leadership positions at Parker Davis, at Aerogenics, Dr. Reddy's Laboratory, and Careus Therapeutics. With more than 20 years of experience in pharmaceutical industry, he has been responsible for leading teams that brought several drug candidates from idea into the clinic. His talk today will be focused on 3D vascular lung model for COVID drug repurposing.
Thank you very much for that kind introduction. I also want to thank Surat and Radha for inviting me. So let me go ahead and begin my speech. So first of all, I want to thank the Academy. Sorry, that's my speech for next week. I'm sorry. That was something else. That wasn't meant for today. But I'm actually going to talk more about the business of science rather than science itself. I come from a heavy industry background. So what I'll walk you through is how the industry uses science. And we have one very bad 4-letter word in the industry. It's not what you're thinking about. It's called risk. So constantly we have to titrate risk in our business. So we do not have the freedom that people in academia have to ask these beautiful questions that you've been hearing about. So we really have to be careful. And as a startup, more specifically, we are bootstrapped. We are working on a minimal budget. So we really have to be careful on what we do and how we spend our money. So I'm not going to show you any data today. If you do a Google Scholar on my name, you will see everything I'm talking about today has been published there. So I'm not going to show you data, but I just want to give you a flavor for how the industry uses science to get products out there, right? In academia, it's about getting publications and grants. In industry, it's all about getting patents and products out there. So a big difference on how academia does science and how we do science. So I just want to showcase for you how we do science. So, right, so the points that I'm going to talk about today are the need for alternate models. I think that's been the mantra from yesterday, so I'm not going to talk too much about that. Alternate models, the status, I think you've had wonderful speakers tell you what the different models are. I won't spend much time. Same with development of 3D models. I want to spend a lot of time on how we developed a human vascular lung model. And again, in our business, the question that is asked is, why did you develop it, right? It's not because I want to get a publication out there. There has to be a business reasons for what we do, and I'll tell you what the reason was. I'm not going to share much data. It's all published. In the interest of time, I just thought that I will not share in data. And then I'll give you a conclusion. I have one slide specifically for the students on, kind of how to navigate yourselves. I think there's also a panel discussion either tomorrow or day after that I'm part of. There we'll talk more there. But if you're already doing your master's and PhD, it's time you started to think about what the next steps are, all right? So this again has been told. The specific purpose I have to show you this slide is that we are in a very expensive business. We go through 15 years before an idea can be translated into a drug. We go through I don't know where this thing is, but drug discovery and development is one chunk of it, which is mainly in the lab. Then you have to go through phase 1, phase 2, phase 3 in the clinical trial. So it's really a huge, huge task ahead of us when we start a project. Not only that, the odds are stacked against us. Generally, 1 in 10,000 ideas will make it as a drug. So we need to come up with thousands of ideas, and very likely only one will be successful. So that's the other thing. And 15 years of work, also the cost. It's very expensive to get a drug out from an idea to a product. can cost you $500 million. 15 years, low odds, $500 million, right? That's like a gambler gambling, and that's kind of what we do on a daily basis. We are rolling the dice and hoping that the dice will fall in our favor. I just thought you should know that. You will hear, besides me, two other experts on drug discovery in the pharma. One is Mani Wangla, who's been asking all those beautiful questions. He's right here. I think he's going to speak on Thursday, right, Manny? You'll hear about more on the development side. I'm going to talk about discovery side. And Suresh Pusala, I want to make sure he's there. Yes, he's there. Is also going to talk about how he's using organoids for drug discovery. So you'll hear about drug discovery. I want to make a distinction about discovery and development. These are completely two different entities. Discovery is a free, you can ask any questions within the boundaries of what the companies you want to do. But development is more regulated. The US FDA or DCGI has very strict rules, and Manny will walk you through some of those. So what I'm going to talk to you about is discovery. So not to confuse with preclinical development, which is a few steps ahead. The other thing in our business we have to do, and the motivation for us to look for alternate to human models. I'm not saying alternate to animals. I mean, we are not here to treat animals, right? So our business is to find how we can mimic humans very early on so we can make the right decisions. Because the later you fail in the business, phase 3, phase 4, by the time you go to phase 2, phase 3, the company probably has already spent $200 million plus. So if you have to fail, the idea is to ask the right questions, use the right models, and fail early in the discovery or early development. That's the whole mandate that we have, is create or use tools that will allow you to make risk-free, solid decision as best as you can. Don't get carried away by the fact that there's a fantastic academic question to be asked. That's not what we are paid to do. Big difference between what we do and the people such as Radha, who have done some very elegant science, are doing. So I just want you to know. So the mantra is fail, but fail early. If you have to fail, fail early. So for that reason, we have to create all kinds of models that are suitable for us. Microfluidics, as you will see, is not suitable for what I do. Does not mean it's not a great model. I think it's a fantastic model, but it has certain limitations from a pharma perspective. That's the reason we do not use it. For one thing, the chips are expensive. You only get one data point per experiment. I do my experiments in a 96 well. I get 96 data points, right? So there are differences like that that are there between academic question asking and industry asking questions. Right, so this again must be very familiar to you. This has been beaten to death. There's clearly differences between how cells look in 2D. They are flat, they're on a monolayer, they can be co-culture, but they're again flat. While as in 3D, they assume the kind of architecture that you find them in real life. And then you can toss in your drugs. The red dots are really drugs. And you can ask any question that you want. So the reason we choose 3D culture is that it mimics human. Again, we have personal no interest in trying to mimic animal models. It's the humans we want to mimic. So the reason we do this is about as close you can get to asking human-based questions without getting into a human. Again, finding ways to de-risk. As I said, you don't want to fail late. If you have to fail, fail early. This, again, is to convince people who are, and Suresh is one of them, who is not convinced that 3D is the right tool. Suresh in the middle is the beautiful pictures of a 3D solid tumor versus the same on a 2D. So enough said, right? So there is clearly, from our perspective, 3D modeling is something more attractive to us. Again, it's horses for courses. Somebody might have a different question and use a different model. Doesn't mean it's better or worse than this. It's just that for our purpose, 3D was the way to go. I think everybody knows why 3D is believed to be better physiologically, or lung cells alone. But really, that's not the true story. A big component of COVID comes from the vessel wall. How the vessel wall reacts to the infection is really a big part of the defense system that humans have for COVID. So we wanted to be able to have a vessel wall and then also the lung cells. Basically, those were the two cell types that we 3D bioprinted, both of human origin. You know about inflammation, so the first thing we had to demonstrate that our lung will respond to inflammatory stimulus and be able to respond to it. So we had certain markers to show that. The other was cytokine storm. I think this morning there was a discussion on cytokine storm. There is absolutely no question that cytokine storm is really a very critical event. In fact, there's now medicines or drugs being developed just to treat the cytokine storm. But before you had any of that, we had the steroids that would be given to the patient. So we wanted our model to have that as well. The third piece that we really wanted to have that nobody else had was thrombosis. Thrombosis, I think, as the previous speakers who spoke about atherosclerosis very elegantly explained, is nothing but the aggregation of platelets in the blood vessel wall. This really happens in COVID. In the young children, in the first phase and the Delta phase that young children died, all died not necessarily because of lung failure, but because they had failure of blood supply to critical organs. This is called Kawasaki syndrome that was seen. So again, to reiterate, none of the animal models, even today, recapitulate thrombosis that we need to see in an actual COVID. So the inclusion of a blood vessel that we included, really that was the whole purpose, is to be able to demonstrate antithrombotic properties of potential drug candidates that we wanted. This is just a picture. Again, if you go to Google Scholar, you will be able to see all of this. At the top, you will see our, the bubble is the air sac cells, airway epithelial cells, then the blood vessel cells. 3D bioprinted, of course, using an inkjet protocol that we developed ourselves. And beautifully, we were able to do this in a 96-well format. So again, going back to saying I can get 96 data points from one plate, because we test many, many drugs, as you will see. At the bottom is a histopathology of the same thing. It looks a lot like human vascular lungs. So it was good enough for us. The COVID relevant endpoints that can be measured in our model were the entry of virus through the binding to the ACE2 receptor. This now, everybody in the household knows about this now. This is what happens. And then the inflammation I talked about. And this we could do by adding exogenously immune cells, such as monocytes, and show the adherence and the penetration of those. And the cytokine storm, as expensive as Karishma said the cytokine measurements are, we did do that, Karishma. So we actually got antibodies, and we set up our own ELISA assays, saved some money there, yeah. So. And then the thrombosis, we were able to demonstrate thrombosis also. These were 4 critical events. So what did we screen? I've already told you that if we went after a new drug, right, it takes 15 years, which means by then COVID would be done and dusted, it would be gone. So that was not the way for us to go, not to look for a new drug. But there are other shortcuts that you can take, de-risked shortcuts. And I'll tell you about that. Yeah. We couldn't afford to do the traditional drug discovery. We don't have deep enough pockets, and all of that. So we had to think of an alternative. So there is another shortcut called drug repurposing. What drug repurposing is if there are drugs already approved by the FDA, you could see if they work on COVID, and if they do, then you can repurpose them. For example, metformin is a drug for type 2 diabetes, right? If you find that metformin can modulate COVID, you can repurpose it for COVID then. That's called repurposing. Why is that important? That's important because it saves about 10 years of time. You can directly go from 3D into clinical trials. So you can get a potentially repurposed drug out into the market in 3 to 5 years versus the traditional 15 years. Also, the amount of money you spend is probably 1/10, right? So that's kind of the reason we had to get into repurposed drug rather than new drugs, because even though our expertise, both Manny and my expertise, is in finding new drugs, we had to take this shortcut simply because of business reasons. So what we did was, this is in collaboration with another very good scientist, Dr. Sridhar Voletti, who runs a company called Indras. He's an in silico scientist. So what we did was we took, as you see at the top of the inverse triangle, we looked at 8,000 FDA-approved drugs. There was no way we were going to screen 8,000 drugs. So he applied his in silico methodologies to select 20 best that he thought, based on the structure, have the potential to either disrupt the entry of the virus. Knowing the RBD and the ACE2 structure, he could do that, or were known to be anti-inflammatory or antithrombotic. So he did beautiful work. It took him about 3 months, and then said, here are the 20 jewels that you really should be screening. So that was our starting point of using those 20 compounds in our 3D vascular model. And we did that, and again, to reiterate, the endpoints in our model was viral entry, inflammation, cytokine storm, and thrombosis. Only those drugs that could, we could check the box for all of this, we thought would be useful in taking further into commercialization. So when we started this, there were no drugs approved for COVID, period. I mean, that was just, not happening. People, physicians, out of sheer desperation, were using drugs that were already there, which were known to be antiviral, right? So remdesivir was tested, which turned out to be a good bet. Chloroquine was tested. Remdesivir, as you know, was not for, it is for Ebola virus originally, not for COVID. So basically, out of desperation, physicians started looking at anything and everything. There was no rational rhyme or reason for them to test it. And initially, chloroquine, as you know, became a big thing. President Trump thought that was going to solve the COVID problem. Unfortunately, it failed. And our model would have predicted that chloroquine would not work, by the way. So we did not want to go the biased way. We just wanted an unbiased way of selecting drugs to be able to screen. That's where the in silico and the 8,000 FDA drugs came. So did we find anything or not? Very interestingly, the first drug we found as a hit that actually checked all the boxes was actually an anti-diabetic drug. This is a drug called, from a series called a target, which is SGLT2. This is a protein in the kidney that reabsorbs glucose, and the anti-diabetic drug basically stops the reabsorption of glucose, so your blood glucose drops. Totally no, out of the blue, we found that this was a drug that actually checked all our boxes. In our wildest dream, we would not have expected this to show up. So that's actually one of the good things about taking an unbiased approach. You yourself don't know what to expect, right? And that's kind of what happened. But very exciting. So did we take it forward into the market? The answer is no. Because once we showed our data independent of us, clinical trials on SGLT2 inhibitors, because they were already approved for diabetes, had already started. And I think one of them is in phase 3. So what it told us was that our model was right, because independent of our work, other people are also testing SGLT2. So we obviously couldn't compete with that, so we actually didn't take it forward, but that validates our model. One of the key things you have to do in the industry and for everywhere else, especially for regulatory people, constantly keep validating the model, right? The example would be, would you allow somebody who is training to be a pilot in a simulator to go pilot a commercial airline? No, right? There has to be a validation. You have to go through thorough training. You have 1 hour ride. It's the same thing in the development part of drug development. Very, very regulated. So, 2 points to make. One is the model seems to work because we picked up SGLT2 as a potential anti-COVID drug, and lo and behold, independent of our work, other companies started doing clinical trials on that. I don't have the final answer on that, but those, I think, trials may be going on. But that was the first win we had. The other family we found, very interestingly, was a series of cancer drugs called tyrosine kinase inhibitors. Again, completely unexpected. We would not have chosen and said tyrosine kinase is going to work. It was purely out of the in silico work that Sri Volette did and our 3D model that picked it up. And so what is the clinical correlate? Did we go forward? Again, the answer is no, because just about when we finished this work, the US FDA announced that baricitinib, which is not listed here, was going to be approved as emergency use authorization for COVID. So very independent from our work. Another validation that the predictive value of our model is actually very high because independent of this work, people have started to move these drugs in the clinic. So that was, again, because it was an already approved product by the US FDA, we obviously didn't want to get into it. So we didn't get into it, but it was very exciting for us to be able to see that, and not one, we picked up a series of, so it wasn't a fluke or a freak. We ran a series of tyrosine kinase inhibitors on the market. Every single one of them actually worked. So there is a mechanistic basis to what we found that, there's a reason why these are anti-COVID. So as I said, in the regulated business, the correlation between the tools you're developing and human biology, really connecting to animal biology is not of much interest to me. You know, we say alternate to animals, but I really think it should be alternate to humans. So SGLT, it had all the properties we want. It's currently in clinical trials, so that's partially a validation of the model. Tyrosine kinase inhibitors that we would have predicted to be could be a drug, has been approved for COVID, so we're not pursuing it. There's one more, there's actually a natural compound called homoharingtonin that we found to be very active, and it's in clinical trials in China. So 3 out of the 3 hit series that we got seem to have excellent correlation with our 3D model. Chloroquine, it was clearly blocking ACE2 binding of RBD, the virus, but it was not anti-inflammatory, anti-thrombotic, and anti-cytokine. So if you had asked us 2 years back, would this be a success? Based on our model, we would have said no. There are other 3D models that we are developing. We have the human vascular lung that can not only be used for COVID, but useful for pneumonia, asthma, COPD, tuberculosis, cystic fibrosis, and other inflammatory lung diseases. So one of the reasons we deliberately chose this model is because it has many applications. You really, in our business, we cannot afford to be a one-trick pony. We can't just have one model and, so we have to have things that can be reused for multiple products. We also have a 3D bioprinted human diabetes model for type 1 and type 2. I'll tell you a small anecdote around that on the next slide. We also have 3D for gut and liver diseases such as IBD, ulcerative colitis, alcoholic liver disease, NASH, and fatty liver. All of this is published. If you Google Scholar, you'll find this. And another one for cancer drug metabolism. We are currently working on 3D human skin model for psoriasis, drug transport, and atopic dermatitis. That's currently under development in the lab. So even though we took a shortcut, right, we could not get drugs out on the market because somebody else beat us to the punch. And this, you have to be prepared. This happens. So what did we do next then? We actually then took another shortcut. So drug repurposing was one shortcut. Our timing wasn't quite right on it. There is another avenue called nutraceuticals. Do you know what nutraceuticals are? Nutraceuticals are common day-to-day nutrients that have pharmaceutical properties. They actually can be used as drugs. A good example would be, I don't know, vitamin D3 right now is associated with so many diseases, right? So vitamin D3 could be a nutraceutical, for example. So that doesn't, just give me 1 second. So you have 15 years for a brand new drug, about 3 to 5 years for a repurposed drug. Nutraceutical can be out there in 2 years. So that's what we decided, that let's just park the drug repurposing on the side. let's go after nutraceutical. Nutraceuticals are gaining in popularity a lot. The reason people didn't go after them before this is because you couldn't get a patent position. But if we have brand new composition and other things, we managed to get a patent position in both. The market is going to be about $75 billion worldwide for nutraceuticals, so it's not small change. It's not 1 or 2 crores. There is a potential to make a lot of money in the nutraceutical business. So that's the shortcut we have taken now. We're using 3D models to go into the nutraceutical business. Nutraceutical has the big advantage, you do not need to do clinical trials at all. FASI, which regulates products in India of nutraceutical, you have to submit an application. If you get approval based on whatever data you said, submit, you can get approval in a month. I think, Manny, we got it in a month or so, the FASI approval, yeah. So you're in the market within a month. That's the approach we took. We said, let's park everything else aside. Let's go use our 3D models to predict what nutraceuticals will work in humans, and let's launch those products. I'm very happy to be able to tell you that we have 2 products launched using 3D. The vascular lung model was used to launch an anti-COVID passive immunization antibody drink. Yeah, you
Dr.
heard it right.
Saxena,
These are antibodies that...
5 minutes.
5 minutes, yeah, I'm done, last slide. You can actually drink. That's been launched in Europe. Unfortunately for us, COVID decided to disappear, so the sales have kind of, flattened a little bit. The other, we went from 3D straight to the market, is something called diabetes. This is for type 2 diabetes. This is not a seasonal disease, so we have now decided to kind of stay away from seasonal diseases. This will actually, this is on the market. You can go on Amazon and buy it today. This is for people who are diabetic and who have been on medication for a long time. A lot of type 2 diabetics, even though they are on every single medicine available, insulin, metformin, glimepiride, GLP-1 inhibitors, GLP-1 analog, everything, they still don't meet target glucose levels, HbA1c, which is the readout. So this nutraceutical actually works on top of all of those drugs. And in very early, in about 15 patient clinical studies, we are clearly able to see that the jump we made from 3D into the humans was actually turned out to be right. Every single patient that we have today has responded with an average drop on top of all of the drugs of 20 to 50%. There's a mechanistic basis for why that's happening. It's not a fluke, so we won't get into that. Immunology also, as I said, went 3D straight into humans. Very small patient data indicates it's also working. So there is a whole new paradigm shift on how new drugs or nutraceuticals are going to be discovered. That 15 years, I think, will have to shrink to about 7, 8 years. And for that, we need alternative tools that have high predictive value. Not high index value of publication, but high predicted value with, this is a point Karishma was making, I think, this morning, and I totally agree with her, is that what you need is predictive value to human disease. That's what the FDA is looking for, and I think that's around the corner with so many very good minds that are working. Do I have 1 more minute, or? Yeah, this is my last slide. I know there is a panel discussion. I don't know when. I think day after. But this is for the students, I think. I'm just going to skip to that slide. I think the MPS that technologies you're hearing today, I think will be of immense interest to the industry, regulators such as FDA and the government. So you guys are lucky to be working in an area that is going to be of critical importance to the industry. You could have easily worked on why elephants don't get ulcers, right? Nobody cares about that. But this is a field that I think really has a lot of job opportunities. And we look for multi-talented people. We'll discuss more. And I think the best way for you guys is to stay in touch with academic and industry key opinion leaders on MPS and just keep following them, and certainly you might have a job. So thank you very much. I'll be happy to take any questions.
Thank you very much, Dr. Saxena. I want to start with a question. Do you think the failure of the drugs is more due to the problems in safety or in efficacy on the development of the candidates?
I think it's, Manny, you can also answer. I think 90% of failure is safety.
Safety.
And that's why you see a lot of times there's a herd mentality in large pharma. So if Merck is following one drug target, shows it to be safe, Pfizer, Sanofi, everybody else will go for the same target because there is safety validation there,
Do we have, well, yes, we have many questions. So. The first in the back.
Yeah, Dr. Saxena, yeah, here. So very nice presentation and actually, very different from, what, we have been listening so far.
Yeah.
Two questions. One is that can you, tell us a little bit about the model? I mean, without hurting your business, but just a little bit about that. And the second is that in case of your COVID-19, that anti-diabetic drug that worked. So, during that COVID-19, that report came, right? That the glycolysis pathway gets enhanced and that, so the deglucose, so that was suggested, which will kind of reduce the glucose content in the cell. So are these two things connected?
I don't know. So the answer to first question, the model is pretty much published. We are not holding, we have filed a patent on it. So after that we published. You can go to Google Scholar, read it up, and I'll be more than happy to have you visit the lab and see the model and do all of that. On the second, to be honest, the SGLT, we haven't spent too much time trying to figure out what the mechanism. The obvious culprit is glucose lowering, as you said, but in the 3D model, there is no glucose lowering, right? So there's got to be something more that's going on. There is certain hypothesis we can do, but we haven't done that. So short answer is we don't know yet.
Yes.
Yeah, I wanted to know, so for your drug repurposing, even if you would have been able to do clinical trials and it would have succeeded, so would you have been able to commercialize that?
Yeah,
and what's the mechanism for that? And what if for that drug somebody else holds the patent? What do you do?
Yeah, those are very insightful questions, actually. You have to be careful in what you choose as a repurposed drug. If it's on somebody else's patent, they will come after you, so you cannot do that. The pathway to get it approved is simply go to the FDA and say, this is the data. The phase 1 data on safety and the phase 2 and phase 3 for diabetes is all there, so safety is not a concern. Please allow us directly go into a phase 3 trial, the last trial, and do it. So that would be the way. So you're saving about 10 years to 12 years of time. Potentially, if the regulator allows, you could be out on the market in 3 years. Yep.
We have also a question in the back with the red polo. Is it? Okay.
Perhaps a very naive question. So what if in a very, imagine this, you have a drug, you know it works, you just don't know how. For this kind of situations, what does a company do? You have every possible, way to prove that it works, just do not know how.
Yeah, the FDA doesn't necessarily need a mechanism, but it's good to know because the mechanism might be tied to a lot of safety concerns, which will pop up later on. So we rarely like to go after, Tulsi extract for that reason, because we don't know what it is doing. We rather focus on knowing that it works through a kinase and the kinase target doesn't interfere with other pathways that can have safety concerns. So short answer is you probably could take it forward, but again, the 4-letter word, the risk is very, very high in doing that.
Hello, sir. Thank you for the talk. I have 2 questions. First is, you told when you were screening for repurposing of drugs, you were unbiased to select the group of drugs you chose to screen. But still, although you are unbiased, but what was the rationale to select even the broad groups of drugs which you chose initially, those 7,000 or 8,000? Then the second question is regarding your anti-diabetic, that Divitis product which you showed. So you told it works above all the other pharmaceutical tablets available. So if there is any fluctuations in the glucose level, whether it is a sudden drop in the glucose or something like that, and despite that, if a patient takes this thing, then how does it affect? Is there any reports regarding that, or what is your
both
comment?
good questions. I will answer the first one. I will ask my co-founder Manny to answer the second one because he's actually using the drug, okay? But sorry, Manny, for disclosing that. But the first one is 8,000 drugs were there. We asked our in silico expert, Sri Voletti, to say, Sri, will it interfere with the binding of two proteins, which is the receptor binding domain of the virus as well as the ACE2. So if you know the structure of both two, you can juxtapose compounds and see there's interference, right? So he shortlisted based on that. Then we said, Sri, are there drugs like this that are known to be anti-thrombotic or anti-inflammatory? So we had filters from the 8,000. Eventually, the filters were designed to be able to act on the properties we wanted to see. So in that way, it was biased, but when we started from 8,000, we had no clue what we'll end up. Second question, I'll let Manny answer. He will answer it only with some sugar and coffee, I guess. I think the short answer is we have not had one incidence of hypoglycemia so far. It's all tied to the mechanism. It's not a fluke or a freak, but Manny will, educate you on that. Yeah.
Sir, here. Sir, in India, why we don't see this industry and academics collaboration? As you said, it takes 15 years to deliver a new drug into the market.
Yep.
So what if industry in India and academics will collaborate and start working? Why don't we see here?
I think there is unfortunately, both were living in their own silos in the past. So CSR labs had a certain mandate to maybe publish and train PhD students. Now the government is mandating more of translational research. So now academia has more of an interest in doing translation work. Otherwise, the elephant and ulcer story was going on for a very long time.
So being a researcher...
Can I just finish my answer? Yeah. So the second part is the industry wasn't ready either, because when we work with somebody, we like to have really neatly written reports, on-time delivery of data. That wasn't quite there, it's coming. So it was on both sides, but that is changing now. Yeah, sorry, please go ahead.
Sir, being a researcher, when we do research in our lab or in any collaboration, we see what is happening inside a cell, what a patient is coming and what we are diagnosing and how we can tackle that. That's the major thing we tackle. I'm from AIIMS, and this is the thing we see in patient reports and try to tackle. And you have a good database where you know what many people know. I'm from AIIMS, I know people coming biochemistry, what they want. But you know what people want from other domains also. So I'm saying if you will come to us, we will collaboratively work and can deliver better things.
Yeah, absolutely, no problem with that.
So recently in AIIMS, we had AIIMS Research Day. This was same question. We had panel discussion for, I think, 1 hour. And DBT director was also there. ICMR director was also there. So still, no, I will say we didn't got any good answer why we are not collaborating. So that's the future. If India wants to progress, we have to do this.
There is no question. And I told you 2 of the reasons which I think are there, but that's going to change. It's going to happen. You need us, we need you. So it's as simple as that, yeah.
Okay, so we have time for one last question.
Yeah, thank you, Dr. Saxena. It's a very useful talk for us. I have a just simple question. When you talk about validation, can you please a little bit elaborate? And is it always necessary to have an intra-laboratory validation? For example, we are developing some model, 3D model of a skin or full thickness, whatever. So is it always necessary to have intra-laboratory validation?
No, intra, I take it for granted that what you're finding you should be able to be reproduced by 500 different reasons. So that's a given. The validation I'm talking about is, let's take 100 diabetes drugs that are out there that have worked in humans and test it in my 3D diabetes model. My 3D diabetes model should exactly predict what was seen in the clinic. So it's called retrospective validation of my model. That's what I was talking about.
Okay. Thank you so much.
Yeah, you're welcome.
Okay, thank you very much.
thank you, Dr. Pamias, for chairing today's session. So that's the end of the conference proceedings for today. We'll break for coffee, and at 4:30, we'll begin the journal club. So I request all of the people who are participating in the journal club to please come back at 4:30. So group 1 can sit over there, group 2 can sit over here, and the journal club will be led by Dr. Girija and Dr. Claudia. So you can go ahead and have your coffee. And at 4:30, we'll also begin day 2 of the workshop. So all of the workshop participants, I request you all gather near the registration desk so that you can head off for today's activities with Dr. Abhijit and his team. Thank you, everyone.